Hello, my name is Ruthie Friedman, and my project is on the quantification of aquatic viruses in hydrothermal vent ecosystems. My PI is Dr. Elaine Luo, and we are part of the Biological Sciences Department. So there's an estimated 10 to the 31st viral particles across all ecosystems, which makes viruses the most abundant entity on Earth. And about half of these viral particles can be accounted for in marine ecosystems. And these aquatic viruses influence ecosystem functioning, biodiversity, biogeochemical cycling, microbial communities, trophic cascades, etc. The list goes on. So the identification and quantification of aquatic viruses helps us understand the extent of their ecological impacts. And viruses in deep marine hydrothermal vents tend to be underexplored. There's not a lot of research out there. So our research focuses on identifying and quantifying these aquatic viruses in a marine hydrothermal vent. And this is done using flow cytometry, which is a standard method for the quantification of aquatic viruses due to its detection sensitivity, reproducibility, high throughput, and characterization of subpopulations. So the hydrothermal vent that we examined was Axial Seamount, which is an active deep sea volcano along the Juan de Fuca Ridge, which is off the coast of Cannon Beach, Oregon. So on this map, you can see the site where here's the ridge, and then here's Oregon, and here's where our volcano is. And axial volcano is characterized by low pH and high hydrogen sulfide and high carbon dioxide levels. And these samples were obtained from 3 sites that were called Anemone, Ashes, and International District. And despite these all being all in the same vent field, each of these sites most likely has different geochemical and physical characteristics. The 3 aims of this research was to obtain viral counts at the 3 hydrothermal vent sites at axial seamount using flow virometry, which is the application of flow cytometry to analyze viruses. We then wanted to observe subpopulations within the samples and compare counts and populations between the sites. Therefore, we hypothesized that viruses will be detected within these samples and that their abundances and subpopulations will vary across the 3 sites due to the variability in these site characteristics. And we also expect there to be about 3 to 4 subpopulations per sample. So the way we did this was 37 samples were taken from axial seamount. They're fixed to a 2% glutaraldehyde concentration and stored at 4 degrees Celsius until they were ready to use. We used a Cytoflex Beckman Coulter flow cytometer, and before samples are run it has to be calibrated using a cleaning solution and DI water, and then you must perform a quality control standardization test, and this just ensures that the noise levels are normal and that your counts are going to be accurate. The samples are then prepared by diluting them in a 1:10 ratio with TE buffer, and blanks were created using TE buffer and ultrafiltrate, which is virus-free water. These samples were then stained with cyber gold, incubated at 80 degrees Celsius for 10 minutes, and then cooled to room temperature before they were run. So the way flow cytometry works is it uses a laser beam to pass through cells, which then scatters light and emits a fluorescence that's detected and converted into a voltage pulse. This data is then collected as scatter signals and can be graphed on a logarithmic or linear scale to understand the concentration of them, as well as like their size, shape, and complexity. And so these samples were run through the flow cytometer for 1 minute at a flow rate of about 30 microliters per minute. And their data was collected in the form of histograms and dot plots, which are seen in these images here. And after the samples were run, the populations were gated, the VLPs were counted, and a statistical analysis, a non-parametric Kruskal-Wallis ANOVA test was run to analyze the samples. So looking at our results, we have it charted here, the minimum, the first quartile, the median, the third quartile, the maximum, and the average counts for each of the sites. So on average, International District had the lowest number of counts with 4.3 times 10 to the fifth. And then Ashes had the most VLPs per mil with 2.3 times 10 to the sixth, and Anemone followed close by with 2.11 times 10 to the sixth. So we can see that there's a decent amount of viruses per milliliter here. And when comparing them, we can see that Ashes and Anemone had a wide range of counts, while International District had a much smaller range of counts, as it was from 10 to the fourth to 10 to the sixth was its top number. So when we ran the statistical analysis comparing these counts, it was found that Anemone and International District were significant with a p-value of less than 0.01, and then Ashes in International District were also significant with a p-value less than 0.001. However, Ashes and Anemone were not found to be significantly different or significantly significant, most likely due to how similar in counts they were, as they're off by just about a decimal place, and you can see their medians kind of line up in the same range, as well as their minimum to maximum range. So getting into the subpopulations, for Ashes, we observed 1 to 3 subpopulations across all the samples. So 8 of these samples had one particular subpopulation, and you can see here on this histogram, we plotted it by count against fluorescence. So this blue here is noise, and then we see this really big clear peak, and this is our first population that we were able to identify. And if you observe it here on our dot plot, we can clearly see 2 significant populations, and with the red being a lot more concentrated. And it's also important to note that while bacterial cells are usually found on the range of 10 to the fourth to 10 to the sixth, we used violet side scatter settings for the flow cytometer, which only allows detection of viral particles because it zooms in on it really close. So these are viral particles, they are not bacterial cells. So continuing with the Ashes subpopulations, 7 of the samples had 2 observable populations. We can see here in the red and green on the histogram, and then also on the dot plot, you can see especially where it's really concentrated, kind of in the middle of these zones that are colored. And then lastly, Ashes was the only site to have 3 populations observed in a sample, and only one of these samples had 3 observable subpopulations. So you can see here in the red, the green, and the purple. And the purple is kind of a small population that's a little harder to distinguish looking just at the dot plot, but because we have the histogram, we are able to identify it as a population. So moving on to Anemone, all of the samples had a single subpopulation. So we can see, as we saw earlier, there's a very distinct peak here that's outlined in red. And then when we move to the dot plot, we can see 2 very distinct populations. And lastly, International District had 1 to 2 subpopulations observed. So 14 of the samples only had a single subpopulation, once again with a very distinct peak and very noticeable difference on these dot plots. And the 2 of the samples had 2 subpopulations that we observed. You can see here that even though they're close together and might be difficult to identify on this dot plot, we can see here on the histogram, there's 2 very distinct peaks. While they are very close in fluorescence, they're still distinct, so we count them as separate populations. So what does this all mean? These numbers and subpopulations have shown that axial seamount is a low viral biomass environment. We only got counts in the 10 to the fourth to 10 to the sixth range, when research has found other marine environments to have upwards of 10 to the seventh to 10 to the ninth counts on average, usually. So this is a very low biomass environment, which means that these are also going to be less genetically diverse. And as you can see, we only saw a maximum of 3 subpopulations, and that was only at 1 vent field site. And research had us expecting to see 3 to 4 per vent site. So these are less genetically diverse. And this probably means that these vent sites have higher percentages of pure vent fluid and higher temperatures. So overall, higher abundances of viruses, typically in marine environments, have larger ecological impacts. The more viruses, the more they can infect and lyse microorganisms, which then put organic matter back into the environment, increasing carbon levels. Higher viral abundances increase the chances of gene transfer, where these organisms could transfer genes and develop into new niches where they can impact other microbes and their trophic cascades and stuff as that. And this also goes into altering their community structure. And higher abundances also increase prokaryotic mortality. So if this is a low biomass environment, that means that this axial seamount volcano is most likely not experiencing these impacts on as large of a scale as some other marine environments are, because it's a low biomass environment. So we might be able to see regular activity within these environments rather than seeing decline due to the amount of viruses. So yeah. And future direction and continuation of this research involves obtaining cell counts to validate the viral counts, because getting a bacterial to viral cell count ratio is really important to also understanding the ecosystem functioning. We also want to cross-validate the data with epifluorescence microscopy counts and comparisons. And then we also want to perform sequencing on these samples to determine their exact viral classification of whether or not they fall under the category of single-stranded, double-stranded, DNA or RNA viruses. Here are my references. Thank you for listening.